Okay, good morning, everyone. Welcome back. Today, we are going to be looking at the effects of dominance and mutation. Right, let me just check that I am sharing my screen. I am good. Just checking I was recording it on Zoom. So yeah, today we're going to be looking at dominance and mutation. So the models that we've looked at so far have been relatively simple in terms of their assumptions and what they've looked at in terms of things like genetic architecture, which is all about the relationships between alleles and loci. So for example, our alleles that we've looked at so far, we've not talked about any interactions between them. So our big A's and our little A's, for example, we've just assumed that, they have some fitnesses and blah, blah, blah. We're going to be thinking about this concept of dominance today, where there's some interaction between those alleles. And also our models so far, we haven't looked at mutation at all. So later today in the second half, we're going to be looking at the balance between mutation and selection and whether it can tell us anything about some real data. Okay, so that's the gist of today. Like I said, in the last lecture, we introduced these diploid models. In those diploid models, we had heterozygotes, if you recall. So these are individuals that have, in this particular one locus model here with two loci, big A and little a, heterozygotes have different alleles at the same locus. So big A, little a, conversely, little a, big A, we were assuming that they were the same thing. And our homozygotes were individuals who had the same alleles at the same locus. So these, remember, in this representation here, the first and the second letters are assumed to be different chromosomes, so one inherited from each parent. So one of the things we assumed towards the end was that heterozygotes had potentially a different fitness to homozygotes, but we didn't kind of discuss why that might be the case. We were just thinking about it in terms of what conditions do we need for there to be a stable polymorphism. So polymorphism, remember, is where we have two alleles coexisting with each other. So today we're going to explore what happens if one of these alleles is said to be dominant and the other is recessive. Okay. Who's heard of dominance and recessive alleles before? Okay. Good. So a few less things to learn today in terms of concepts, but we'll be focusing a lot on this idea of dominance and recessive alleles. We're going to be talking a little bit about when there isn't dominance, and so we have what's referred to as additive fitness effects. And then, like I said, we'll be focusing towards the end on the balance between mutation and selection. Okay, let's start off. We say that an allele, A, big A this case, is said to be dominant if the heterozygote that contains these two alleles, big A and little a, has the same phenotype as the homozygote, big A, big A. Can anyone remember what phenotype means? Yep.
It's like the physical expression of that.
Exactly, yeah. So, for example, you might have a phenotype that is blue eyes or brown eyes. We have various different phenotypes. But, a characteristic or a trait that is expressed as a result of a gene. It's also usually interactions with the environment as well, but we're going to simplify that and just focus on the genetic effects here. So there will say be an allele that encodes for certain traits, and that's the genotype. But then the actual phenotype is what is expressed. Okay, so what you see, if you like, what an organism expresses to the environment. Okay, so an allele big A is said to be dominant if the heterozygote containing both of these alleles has the same phenotype as the homozygote with the dominant allele. And then the little a allele is therefore said to be recessive. Okay, so it's not possible for both of these alleles at the same locus to be dominant. One of them would have to be dominant, one would have to be recessive by definition. Okay, so we still have our homozygotes with both little a alleles expressing the little a phenotype, but in this case, our homozygote and our heterozygote have the same phenotype. So you won't be able to tell them apart unless you looked at their genetics. Okay, so a simple question to get us started and get you thinking about this. Suppose we have a population that only consists of blue and brown-eyed individuals, and that mating occurs at random. This is a large population, so we can treat things as being deterministic, and we don't have to worry about there being sort of random effects. Also, there are no differences in fitness, we're going to assume, between blue and brown-eyed individuals. And we're going to assume as well, I haven't put it on here, but we're going to assume that the alleles encoding for brown and blue eyes have been in the population for a really, really long time, so that they've both kind of settled down to 50-50 frequency, okay? So they're both at equal frequencies in the population. So what I want to know then is, what is the expected frequency of each phenotype in the population? And then secondly, if both parents have brown eyes, what is the probability that their offspring has blue eyes? So have a go at that for a few minutes. Chat to the person next to you as well. You don't have to do this in silence. It's not an exam. And work through those problems, okay? And this is just to get you guys thinking about what it means for things to be dominant and recessive. Okay, let's go through this one. So I'm going to get Mark to do this, if you'd like to. So just starting off to discuss, like, one of the key assumptions here that isn't written down but I said out loud was that we're assuming these alleles have been in the population for a really long time. And because there's no fitness effects or fitness difference between them, you've essentially kind of just got a random walk. So over a long period of time, they should settle down to roughly equal frequency. Okay, so essentially we're assuming if we let P be the frequency of, say, the brown-eyed allele, I'll call this capital A, then P is equal to a half. Okay. What else are we going to assume here if there are no fitness effects? We went through it last time, had a particular name for a type of equilibrium. It tells us something about the distribution of the different genotypes in the population. It is? Yeah.
Is it Fisher-Haldane?
Close.
Oh,
It's one combination of names.
Fisher-Haldane, right?
Not that one, it's the other one. Hardy-Weinberg, yeah? So we're going to assume Hardy-Weinberg equilibrium. Okay, at that point, I'm going to hand over to Mark.
So for, I'll go start with question one here.
Yeah.
So, for question one, we have an equal, proportion of each, allele. So we have,
You got to play like the lottery.
when we're passing the genes on, we have, the four possible, or sorry, the two possible genes from each parent, right? So either we get A little a, big A, or little a. And since the big A is dominant, we're gonna have, these will both have brown eyes, and of course, this one's gonna have brown eyes. The only one with blue eyes will be here. So we'll get a 75% proportion of brown eyes, and 25 blue eyes, assuming that they're equal, each of these are.
Perfect. So can anyone remember what this kind of diagram was called? Punnett square, exactly. Yeah. So it's really helpful, like Mark's shown here, just to think about all the different combinations to make sure you don't miss anything, right? So because there's no fitness differences, P and Q are at equal frequencies, we're just gonna have a quarter in each of these boxes. So when you sum it up, you end up with 3/4, 75% have brown eyes. And that's the brown-eyed phenotype. So that's the first part. Hopefully that was relatively straightforward. How about the second part? Why don't you explain your reasoning
So
for
for
that?
the second part, we're considering two brown-eyed parents. So first, let's consider all possible combinations of brown-eyed parents. So from here, we already saw, we have A little a, or little a, big A. And for the second parent, it's the same thing again. A little a, and little a, big A. And then in order to, or now we're gonna look at the different combinations. So each parent could pass on either this one or either this one, right? And then same as this one here. So for each of these, we're gonna have 4 different combinations, right? So we can have like this one and this one, this one and this one, and so on, right? So for the first one here, we're never gonna get a little a, right? Because we only have big A's. So we know that we're just gonna get 4 different combinations of big A's here. Here, we really have the same thing for this whole row and this whole column. Because as long as we have one little a, or sorry, one big A, we're always gonna have the brown gene, right? So for here, we'll get like AA, 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 AA. Here, we get a similar thing, AA, A little a, AA, and A little a, and so on and so on. And we can see that each of these here is gonna have 4, and the only possible way to get 2 little a's is gonna be this guy and this guy, this guy and this guy, this guy and this guy, and this guy and this guy here. So we can see that's 4 possible ways to get 2 little a's, and we have 4, 8, 12, 12, 12, so 36 possible like children, I guess, so to speak, so 36 possible combinations, and only 4 of them can be little a. So 4 over 36 is our ratio for blue-eyed, or our like percentage of blue-eyed. Yeah.
Aren't we overcounting because some of them are the same? Like
Um,
AA and AA.
like, what do you mean?
Like in some of the groups, we have like 2 big A's and 3 big A's.
So the way that I'm looking at it is, so we have a parent with 2 big A's, right? They can pass on this big A or this big A, right? So if they pass on this big A and this parent pass on this big A, that's one combination. This big A and this big A is a different combination,
Oh.
and same as this one and this, right?
Yeah.
And then this one and this one again. So that there's 4 different combinations for like each of these, because you have like 1, 2, 3, 4, right?
Okay.
And even though like you still have like big A, it's a different gene, it's like a different part, I guess, of the or a different allele getting passed on. So it is a different combination.
Okay, thanks very much. Yeah,
And I have a question for you.
of course.
A lecture or two.
Thanks very much,
back,
Mark.
I'll tell you so-
Sorry. Thanks, Mark.
A lecture or two back, you said that that we can treat big A, little a, big A.
Yeah, this is what Mark's done here is just kind of like thinking, although these are in some sense just like different labels, they're phenotypically the same individuals, and so that's why we treated them as having the same fitness when we were doing those diploid models. You can structure out this to think about every possible combination if you think about like, because of the manner that these we treat these guys the same in terms of all their fitnesses, but essentially there is a different genotype depending on which parent they've inherited it from. We just say that they don't have any differences between those individuals. So all that Mark's done here is kind of think about all possible combinations. So the other way of thinking about this is that our brown hetero. So as Mark said, the only way you're going to end up getting a we're told that both of our parents have brown eyes, but we don't know if those parents have are homozygous or heterozygous. All we're told is that they have brown eyes. So what we need to think about is, okay, what's our frequency of our brown homozygote? Well, that's going to be individuals who have AA. And so we know from above that they're going to be a quarter of individuals in the population will have this genotype, big A, big A. And the heterozygote, well, those are going to be big A, little a, or conversely, little a, big A. And those are going to be half of the individuals in the population. Okay, so that's just these squares here, right? So it's all the brown individuals, people who have brown eyes. So if we're told that both of our parents have brown eyes, what does that mean? Well, 2/3 of individuals that have brown eyes are heterozygous, right? So half of the population is heterozygous, but 2/3 of individuals, so these 2 boxes here, 2 out of these 3 boxes are individuals that are heterozygous, right? So we say have 2/3 of brown-eyed parents are heterozygous. So probability, that's 2/3 of brown-eyed individuals really, rather than parents. So probability that both of these are heterozygous is just going to be 2/3 squared. 2/3 of brown-eyed individuals from whom we're choosing our parents are going to be heterozygous. So that's 4/9. And then the probability that our offspring is homozygous for the recessive allele, given that both parents are heterozygous, it's just going to be 4/9 times by what? 1/4, right? Because, as Mark said, these parents, I mean, it's essentially this similar to here. There are 4 different types of offspring that they can produce between these 2 individuals. I say 4 different types in terms of the order of alleles. Sometimes the ordering doesn't matter. And the only one that we care about is when they're both little a. So it's 1/4 of those because there's no biasing here at all. So we end up with 4/9 times 1/4, which is a 9. Okay. Yes.
Why did we not? I don't see how we didn't account for the homozygous in case of the parents having brown. Like, isn't there a combination with this with brown eyes, like AA and then little a, big A?
Because the only way that we can have a child that has blue eyes is if they inherit a little a from both parents.
Oh, okay.
So the parents have to be heterozygous. If one of them is homozygous, the children are always brown-eyed. Yeah. Okay, let's move on. We spent a little bit longer on that than I pretty anticipated, but it's okay. We've got quite a bit of time today. So recall the Fisher-Haldane-Wright equation from last lecture, and this tells us about the change from one generation to next of the frequency of a particular allele. So it was written in this form here, which I won't read out, but it's on the slide. And if you just recall that P is the frequency of our big A allele here, Wij is the fitness of the genotype with alleles I and J, and W bar is the mean fitness of the population. And if you recall, we also discussed at the end how this could be written in a slightly different form. Where PT plus 1 is equal to P of T plus P of T times by Q of T times by this fraction here, which is the difference in fitness between individuals that are homozygous with the big A allele and heterozygous individuals times by the frequency of the big A allele. And then similarly, the difference in fitness between heterozygous individuals and individuals that are homozygous for the little a allele times by the frequency, and then divided by mean fitness. Well, we didn't go through how we get from one of these to the other. That was left as an exercise. But these 2 things are equivalent. And sometimes it's easier to work with this formulation. Sometimes it's easier to work with this formulation. Turns out when we're looking at the case of dominance, it's easier to work with a second formulation. Okay. So let's think about selection for a dominant allele. So we're going to suppose again that our big A allele is dominant, and we're going to suppose that it is advantageous. So there is a fitness advantage of having this big A allele relative to the little a allele. And it's dominant, so we can write down our fitnesses. If we suppose, just for simplicity, that our homozygous individual with the recessive allele is equal to 1, so W little a is equal to 1, then W big A is going to be equal to 1 plus S, where S is our selection coefficient. And what about the fitness of the heterozygote? What's this going to be equal to? Anyone? So our homozygous individual with the recessive allele has fitness 1. Our homozygous individual with the dominant allele has fitness 1 plus S. So what's the fitness going to be for our individual who's heterozygous?
Just S.
not just certain. Remember, if the individuals have the same phenotype, right? That's what it means for it to be dominant, right? So an individual has both sets of dominant alleles, it's going to have the same fitness as an individual who has just one of them, right? Because it's dominant. So this is just going to be 1 plus S as well, right? They're identical individuals. Think of this as just being individual blue eyes, individual brown eyes, and we're supposing there's a fitness difference between individuals who have blue and brown eyes, right? But these individuals both have brown eyes. So they have the same phenotype, they have the same fitness. Okay, so what about our mean fitness then? Well, our mean fitness, remember, it's just going to be if we're assuming things are at Hardy-Weinberg equilibrium, it's going to be the frequency of each of these different genotypes multiplied by their fitness, okay? So the expected fitness, the average fitness here is going to be the fitness of our first genotype, big A, times by p squared. Is the frequency of allele big A. And then we have a 1 plus S times by 2pq. That's the frequency of 2pq is the frequency of our heterozygous individual, and it has fitness 1 plus S. And then we have 1 times by q squared. So that's the fitness of our homozygous individual with a recessive allele times by its frequency q squared. And should hopefully be relatively straightforward to see. We're going to have, because of these ones in here, so we've got a p squared, a 2pq, and a q squared. That factorizes to p plus q all squared, so that's just going to be 1. So we end up with a 1 out the front, and then we're going to have s times by p squared plus 2pq. I'm just going to leave that in. Obviously, I could factor out a p here as well, but I'm just going to leave that there, just so it's clear where it came from. Okay, so what happens then to allele big A in the long term if we're starting with both alleles present in the population? So our initial frequency of P is not 0 or 1. If both alleles are present in the population, we're assuming this because we're assuming there's no mutation. Otherwise, we would just stay wherever we stayed at. Okay, we started at. Okay, so if we go back to our Fisher-Haldane-Wright equation up here, we have all of the things we need to write down a difference equation for selection for a dominant allele. Okay, so just copying what we had above, P of t plus 1 is going to be P of t plus, we had a PT QT on the outside, but I'll just pause on that for a second and think about what was in this fraction over here. Well, because our big A allele is dominant, that means what? The fitness of these two guys are the same. So the nice thing about this format is this first term here just goes to 0. And in the second one, we had the fitness of our heterozygote is 1 plus S, and then we're going to be minus 1 because that's the fitness of our homozygous individual, the recessive allele. So we just end up with S times by QT from that. So taking into account the P of t and QT on the outside of that fraction, we end up with S PT Q of t squared all over our mean fitness, which we've just worked out below. We have 1 plus S times by P of t squared, P of t squared plus 2P of t Q of t. Okay, so what can this tell us then? Well, all the terms here are positive, right? So S, we're assuming, is a positive selection coefficient. Everything here is positive. That means that since, just write it out, since S is positive, the second term, the fraction is positive. So P of t is monotonically increasing. Slight caveat to that, when I say this is positive, this is initially positive at least. Why might it not? It can never be negative, but it can be 0, right? Because if P of t equals 1, then Q of t is going to be equal to 0. But until that point, it is positive and it's monotonically increasing. P is bounded by 1. bounded above by 1. So P of t tends to 1 as t tends to infinity. So we have selection for our dominant allele. It's advantageous. That shouldn't be too surprising. Something that's advantageous to spread into the population. But now that we've got this format here, we can ask how fast does this dominant allele spread when it's relatively rare? So we can think of this as being a case where, say, our recessive allele is actually initially just the only allele that's in the population, and then a mutation arises for an allele that happens to be dominant, and that is also advantageous. So how quickly is it going to spread through the population when it's initially rare? That's kind of the question we want to answer here. So what we're essentially assuming is that P of t is close to 0, so we can approximate this difference equation above. We're saying P of t plus 1 is approximately equal to P of t plus, well, on the top, S is just a constant. P of t, we're assuming is very, very small, and we've got a Q of t squared. Q of t is going to be very close to 1, so we can approximate this as being approximately equal to 1. So we're just going to have S P of t on the top. And then on the bottom, we've got 1 plus, if we were to take this P of t outside, we'd have S P of t times by something, right? And the P of t is going to be relatively small. So this is approximately equal to just 1 on the bottom, because we've got 1 plus something very, very small. So it's roughly equal to 1. So this works out nicely as just being approximately equal to 1 plus S times by P of t. This means that the growth rate, when it's relatively rare, is approximately exponential. That's because we can write down P of t is equal to P of 0, our initial condition for frequency of the big A allele, times by 1 plus S to the power of t. Right. If you're not familiar or haven't seen a difference equation solved like this, it's because we can write P of 1 is equal to 1 plus S times by P of 0. P of 2 is then going to be equal to 1 plus S times by P of 1, which is just 1 plus S squared times by P of 0, and so on. So this is exponential growth, and exponential growth is likely to be quite fast. Okay, so that's a dominant allele. What about if our advantageous allele is actually recessive? And so we're still going to assume it's advantageous. In this case, we're going to assume that WAA is equal to 1. And this time we're going to have, again, our homozygote for this advantageous allele is going to be 1 plus S, just like we had before. What's going to be the fitness of our heterozygote now?
1.
1. Why is it going to be 1?
Because it's the same as the little allele.
Yeah, exactly.
Because big A is recessive.
Okay. So I'm sure I'll work through this one. So again, we're going to determine how A grows when it's relatively rare, and how does it compare to the growth case scenario when big A was dominant. Okay, so let's work through this here. First of all, we need our mean fitness of the population, because that features in our Fisher-Haldane-Wright equation. So just like before, we need to take the fitness of each of our genotypes and multiply by its frequency. So we're going to have 1 plus S can be multiplied by P squared. So that's the fitness of our homozygote. I misspoke, I realized earlier before. I said that we were assuming that these were in Hardy-Weinberg. I mean, they can be at any. Well, essentially, yeah, we have, after random mating, you would end up with these frequencies P squared plus 2PQ plus Q squared, but they don't have to be, they're not staying at a fixed level because the fitnesses are going to be different, right? So it's not it's not an equilibrium in that sense. Okay. So if 1 plus S, our heterozygote is going to be having the same fitness as our homozygous individual for the Lille allele, and this is just 1. So 1 times by 2pq, and again, plus 1 times by q squared, because that's the fitness and frequency of our individuals who are homozygous for the Lille. And then this, again, nicely cancels out in a similar way to before. We end up with 1 plus p squared times by s. And this 1 comes from the fact that we're going to have a p squared plus 2pq plus q squared, which simplifies down to p plus q all squared, which is just 1. Okay, so let's go back to our equation for the change from one generation to the next of our big A allele. So from our Fisher-Haldane-Wright equation, I'm not going to write it out again, actually. What I will do is I'm just going to copy it from up here. A little bit quicker. So this is just our Fisher-Haldane-Wright equation. And as before, we've got all of the things we need to substitute into here, right? So we're going to have, again, this P of T out the front. I'm just going to jump straight ahead to think about what's going on in this fraction and leave the terms outside for a moment. So now there is going to be a difference in fitness between these two types of individuals here, big A, and big A, little a. We're going to have 1 minus 1 plus S. Oh, no, sorry, it's going to be 1 plus S minus 1. Sorry, getting ahead of myself. So we would have this term here, just highlight it. We can have a 1 plus S minus 1, so this is just going to be an S times by PT. And then here, these things are going to be the same. So the fitness of these two guys are the same because our big A allele is recessive. So the second term goes to 0. So we're going to have an S times by P of T squared, that's by Q of T over our mean fitness, which we just worked out to be 1 plus P of T squared, that's by S. And we want to do exactly the same as what we did before. We want to think about what happens when P is relatively small. This is when our big A allele is rare. So how does it spread when it's rare in the population? In other words, when P of T is approximately to 0, so when just P is close to 0, we can approximate this equation above. P of T plus 1 going to be approximately equal to P of T plus, well, on the top, just as we had before, our Q of T is going to be very close to 1 because P of T is very close to 0. So it's going to be approximately to S times by P of T squared. And then on the bottom, because P of T is very close to 0, 1 plus P of T squared times by S is also going to be very close to 1, right? So everything cancels down to 1. So again, this is the nice way, the nice thing about using this format of the FHW equation. It ends up cancelling lots of things out, and then we can make some nice approximations to growth when things are rare. So then this just simplifies down now to P of T times by 1 plus S times by P of T. Okay. So what can this tell us then about the growth of a recessive allele versus a dominant allele? Well, these two things were quite similar. This thing we ended up with here is quite similar to what we had up here, with the exception here, we just had a 1 plus S, so this is just a constant. Whereas down here, we had 1 plus S times by P of T. So can anyone think about how the growth rate in this scenario is going to compare to the one before? Is it going to be faster or slower when P is rare? Slower, right? Because P of T is less than 1. So we can say that this is slower than for a dominant allele. Intuitively, what's going on here is that when our big A allele is relatively rare. Most of the time, it's finding itself in heterozygous individuals, where there is no fitness difference, right? Because it's recessive. So most of the time, early on, even though it's advantageous, it's very rare for it to be in a homozygous individual, big A, big A. Most of the time it's in heterozygous individuals, so it's going to spread really slowly. Whereas when we had these homozygous individuals, when it was dominant, our homozygous and heterozygous individuals have the same fitness. Again, it's still finding itself most of the time in heterozygous individuals when it's rare, because little a is most common. So when individuals mate, they're most likely to be homozygous for little a. But it doesn't matter there, because heterozygous individuals have the same fitness as the homozygous. So whether something is dominant or recessive has a big impact on when it spreads when it's rare, even if it's advantageous. Again, something relatively profound that we can learn from these relatively simple models. Okay. We're going to go a little bit more, and then we're going to have a break. Just want to get up to stuff about mutation selection balance, and then we'll do that in the second half. Okay, so what can we deduce then about selection against a dominant or recessive allele? We've talked about selection for an advantageous dominant or recessive allele. That's all we've done so far in these two different scenarios. What about selection against? So now the allele that we're thinking about is no longer advantageous, but it could be dominant or recessive. Anything that we can conclude based on what we've just learned from selection for dominant or recessive alleles? Any ideas?
Selection against a dominant allele, there will be like reduction in the allele frequency.
Yeah. So think of it this way. Okay, so because of the symmetry here, if I say you're not wrong, selection against a dominant allele is going to be similar to what?
Selection for recessive.
Exactly. So selection against a dominant allele is going to be exactly the same scenario. We're just kind of flipping things around, right? To selection for a recessive allele, and likewise selection against a recessive allele is going to be like selection for a dominant allele, right? That's exactly what we've just modeled here, right? Like we've framed everything in terms of selection for a dominant allele, selection for a recessive allele. But if you think about things from the perspective of the little a allele, it's selection against that. We've talked about, when things are common versus when things are rare. I mean, we can think about, again, the scenario of a rare big A allele spreading into a population that's advantageous. That's exactly the same scenario as thinking about our little a allele being selected against. So the nice thing here is that we can just use exactly the same scenarios that we've looked at already to understand these things. So briefly, selection against a dominant, I'm going to put in brackets recessive, just so that I don't have to write this all out twice. Allele is the same as selection for a recessive. Yeah, in brackets dominant, so thinking about the other way around. Allele. So when I put these things in brackets, it's just think about the difference. So selection against a dominant allele is the same as selection for a recessive allele, and also selection against a recessive allele is the same as selection for a dominant allele. Okay. The last thing we're going to do in this first part of today's lecture is thinking about what happens if there's no dominance. So we've just considered today these examples where big A allele is dominant or recessive. But if neither allele is dominant or recessive, then we refer to this as usually as having additive effects. Okay, so what this means in terms of our fitness of our different genotypes, if big A allele is advantageous, then the homozygous individual here is going to have the highest fitness. Our heterozygous individual only has one copy of that allele, so it's going to have intermediate fitness. And our individual that's little a has no copies of that advantageous allele, so it has the lowest fitness. And then conversely, if the big A allele is disadvantageous, it's just going to be flipping those signs around, those inequalities around. So our heterozygous individual in this case has intermediate fitness. Based on what we learned last lecture, are we going to have stable coexistence here with these alleles? Yeah, it is. No, I'm seeing a few shaking of heads. Yeah. Do you remember that when we proved that by looking at this version of the Fisher-Haldane-Wright equation that has stable coexistence, these two terms here are going to have different signs. And that's not going to be possible if our but this is only possible, sorry, if our heterozygous individuals are both either more fit or less fit. They either have to be superior or inferior to both homozygous individuals. Okay, so we've got no dominance here. Intermediate fitness. This is our additive scenario. We could do a similar process to before, though. So suppose now that each allele A has equal effects on fitness. So because they're additive, having one copy of the allele has a fitness effect of S, and then two copies is going to be 2S. So our homozygous individual for big A is going to have fitness 1 plus 2S, and the heterozygous individual is going to have fitness 1 plus S. And we're assuming that our homozygous individual for little a has fitness of 1, just as like a reference point. And as before, we can work out our mean fitness just by thinking about the fitness of each of these different genotypes times by its frequency. So 1 plus 2S times by p squared plus 2 lots of 1 plus S times by pq, and then plus q squared. As before, we're going to have 1 times by p squared and 1 times by 2pq plus a q squared. This all simplifies down to 1 for the same reasons as before. So I have a 1. And we're going to have 1 plus 2sp times by p plus q. It's because we had a 2sp squared and a 2spq in the first and the second terms. And p plus q is just equal to 1, so this is just 1 plus 2sp. Okay, we don't actually have to worry at this stage about whether this big A allele is advantageous or not. It's just a slight typo in the notes there. Should say for some s greater than or equal to minus a half, because we're going to assume that it's not going to cause fitness to be of this individual that is has both copies is going to be positive, or at least non-negative. So for some s greater than or equal to minus a half, our FHW equation, we just write in exactly the kinds of things that we've done before. Okay, so P of t plus 1, P of t plus P of t, Q of t over our mean fitness. I've just left that in there for now, just so we can focus on what goes on in this big bracket here. A little bit more complicated to before. This is because we're going to have a 1 plus 2s minus brackets 1 plus s. So that's the difference in fitness between our homozygous individuals with big A and heterozygous individuals. That's going to get multiplied by P of t. And then fitness of our heterozygous individuals, 1 plus s, minus the fitness of little a homozygous individuals, which is 1, times by Q of t. So what happens here? We are going to have a 1 minus 1 in this first bracket, so the 1s are going to go. Out, so we have the 1s are going to go. We have plus 2s minus s, so we're just going to end up with plus s. from this first bracket. In the second one, we're gonna have the ones cancel as well. So you end up with a plus S times by P of T and plus S times by Q of T. So we're gonna have S times by P plus Q. P plus Q is just equal to 1. So all we get out of this is just an S. We have S, P of T, Q of T, and then we had our mean fitness on the denominator, which is just 1 plus S times by P of T. Yes?
Wasn't it 1 plus 2s on the bottom?
Yes, sorry, I've missed a 2. It was in my notes, but I took it probably down. Yeah, thanks. So 1 plus 2s P of T, thank you, on the bottom here. Allow you guys to write this down. This is in the notes in case you don't actually copy it all down. Okay, so then lastly, what is the approximate growth rate? Then, just like we've done before with our dominant and recessive scenarios, when our big A allele is rare. So when P is approximately equal to 0, we're going to have P of T plus 1. It's going to be approximately equal to P of T again, plus, well, the same logic as we had before. Q of T is going to be approximately equal to 1. And then on the bottom, we're going to have 1 plus something very, very small. So that's going to be approximately equal to 1. So this is going to be approximately equal to P of T plus S times by P of T over 1, which is P of T times by 1 plus S. This is just the same as we had before, right? So when it's very, very rare, it grows at approximately the same rate as when it is when it's dominant and when it's additive. All of these approximations, just remember, these only apply when our big A allele is rare. So as soon as we start moving away, it starts increasing in frequency. This is going to differ, basically. We have assumed, I mean, I said above that we're assuming that S is greater than or equal to -0.5. When we're assuming in this scenario here, this is an advantageous allele A, so S is positive in this scenario. So the general form above here was for any realistic S. Here we're just looking at an advantageous one. Obviously, if it's disadvantageous and it's rare, it's just going to be selected out of the population. So it's not very interesting. Okay, we're about halfway. So let's have a break for 10 minutes. We'll come back about 25 to 12. And I'll stick around here if anyone has any questions in the meantime. Okay. Okay. The second half, I think, should be a little bit shorter, actually. So there are some exercises at the end that you can hang around and do, but also can kind of leave it early. I'm a little bit loath in these 2-hour sessions to like just lecture at you for 2 hours, because there's going to be a limit to how much you guys can absorb. There's a limit to how much I could absorb if I was sat in your place as well, right? So I'll try where possible, especially in the second part, to do a few more exercises, or at least have them interspersed across, so you're not just kind of listening to me for 2 hours. Couple of announcements as well before I forget. So I did send an announcement out on Canvas. I managed to change the room starting from next week. I reminder as well that Monday's a public holiday, so there's no classes. So next Wednesday will be in AQ 5018, if I remember rightly. So in 5th floor on the AQ. So there, hopefully there should be a projector in the middle, so you guys won't have to strain over to the corner. And also the first homework assignment is now online as well. If you have any questions about that, or if you think you spot any mistakes, hopefully there won't be, but I'm only human. So if you spot anything that you think doesn't look right, please drop me an email. That's due next Wednesday. Again, if any of the instructions aren't clear or whatever, please just feel free to reach out. I'd be happy to clarify anything. And there is some like coding aspects to this, but I say that in a very loose sense. It does state on there, if you want to use something like ChatGPT to help you write code, that is, for the solutions, that's how I've generated the solutions. So it is possible. I would be slightly cautious that the output of ChatGPT or similar tools doesn't always give you the correct thing. So don't just assume that what your prompt is the correct thing. You might have to adjust things yourselves. I believe for this one as well, at least especially for the undergrad part of it, that you could do it in Excel if you really wanted to as well, right? Because it's all about difference equations, so you can plot things in Excel. So if you don't want to do the coding side, that's fine. Other assignments you probably will have to do at least some coding. Okay. If you haven't used Jupyter Notebooks or something like that before, you can access like Collab Google. It's free, and also there's SFU Access. I've posted those on the homework assignment page as well, those links if you've not used them before. And I think that's it in terms of homework. Just a reminder as well, actually, at the last part of the homework, grad students, you have an additional component to that. Undergrads, you can get bonus marks if you do that, but obviously you don't have to. It's just there for bonus marks. Yes.
So I have two questions. Do we have to use Jupyter or whatever? Can you code with?
You can code in another language if you want. Like, to be honest, I don't care what language you code in, or even, you know, like I say, if you used Excel for the particular problem in today's homework. I care more about you being able to solve the problem, not the method that you use to solve the problem. Okay.
And then what will that mean for all stars next week?
That's a good question. I don't have any particular office hours next week because of the holiday, but what I'll do is if you have particular questions or concerns, just email me and I'll be happy to answer those, or I can arrange a time to meet on Zoom or something like that. So yeah, the Monday makes it a little bit tricky with my schedule, but do feel free to reach out if you guys have questions. Okay. Well, I'll say don't reach out at like Wednesday at 11 p.m. asking for questions on the homework, because I probably won't respond. Okay, so second part of today, we're going to be talking about mutation for the first time and the balance between mutation and selection. Again, this is a very fundamental thing that occurs in populations. Everything we've been assuming up to now is there's been perfect inheritance. So offspring inherits the alleles from their parents perfectly. There's no chance of any errors. Of course, that's a great simplification. In real populations, we have mutation. And in fact, if we didn't have mutation, then we're not going to have the sort of raw material to be supplied for selection to act upon, right? So mutation isn't necessarily a good thing. Why do we have mutations? For example, when DNA is replicated, there are errors in that process. It's not a perfect process. This can cause our alleles essentially to change from one to another. There can also be sort of mutagens as well in the environment that might cause these errors, or can cause other things like breaks in DNA, and then they come back together again, and then they're not quite recombined perfectly, or not reformed perfectly. Okay, so the other thing is that as mutations are only passed through the germline, don't really worry about this as a terminology. I usually bold like biology terms if you want to look them up more. You don't need to know the germline, but we have two types of cells essentially. We have our normal tissue cells, and these are referred to as soma. And then our germline is essentially the sex cells that are passed on, right? So, if you have mutations that occur in your arm, that's not going to affect what's being passed on to your offspring. It only matters in terms of your germline, so what's affecting your gametes. Okay, so we don't care about mutations that are occurring elsewhere, just what's going on in terms of inheritance. Okay, that's kind of the background. And the goal here, when I say this mutation selection balance, is that in the models that we've looked at so far, if an allele was advantageous, it would ultimately just go to fixation, right? We've been talking about dominance and recessive, and ultimately thinking about the rates at which things are changing when they're rare, but everything ultimately in those models we've looked at today, if it's advantageous, it will eventually reach fixation. However, if we've got mutations, you can think of that as a force pushing you away from that fixation, okay? And the more mutation you have, the more you're going to be pushed away from that. So you've got mutation pushing you in one way, away from your equilibrium, if you like, or your steady state, but you've got selection pushing you back. So you've got these two opposing forces. And then we want to think then about the balance of these two forces. Can it tell you something about the frequency that you'd expect these alleles to be at? Can it tell you about the rates of mutation as well? So let's think first of all then about a slight adjustment to our model. So we're now going to assume that our big A allele is dominant and our little a allele is going to be rare and deleterious. So it's a slight switch around from before. just because it slightly helps a little bit of notation. So our big A allele is dominant now, and it's also the most common thing. And our little A allele is rare and it's deleterious. So in the notation that we had before, switching things around a little bit now, but we're going to assume that the fitness of our individuals with at least one copy of the dominant allele is going to be equal to 1. And individuals with the little a allele are going to be having fitness 1 minus s, where s is some positive number. Okay. So if you recall in the last lecture, we introduced some definitions for the fitness of individuals with these alleles. So with allele big A or little a. So just to make sure this is really clear, these fitnesses that I've just written above are the fitnesses of genotypes. So individuals that have big A, big A, big A, little a, little a, little a. Okay, so these are individuals who have these genotypes, but then we can average over individuals that have copies of these genotypes to work out what is the fitness of that allele. Okay, so individuals that, this is composed of individuals that have 2 copies of that allele and 1 copy of that allele in terms of big A, and likewise, individuals with little a will have 2 copies in homozygous individuals, 1 copy of the heterozygous individuals. So we defined this in the last lecture. But it turns out that we can write these then, the fitness of individuals with big A allele are going to be ultimately P plus Q times by 1, which is just 1. And for our little a allele, we can have 1 times by P plus 1 minus S times by Q. This is just the frequency. This ultimately comes from the frequency of the heterozygote and the frequency of the homozygote when things cancel down. So this is just going to be P plus Q minus SQ, which is just minus SQ. I'll write it as QS actually, for no reason other than alphabetical. Okay. So we also talked before about the selection differential, and we can write this now in terms of our little a allele. So little a allele, its fitness, minus the fitness of the big A allele relative to the big A allele. So remember before we had this round the other way, big A allele minus little a allele over little a allele. Now we're just writing it the other way around, yes.
I thought P plus Q was 1, so isn't it 1 minus QS or not?
Yes, it is. Sorry, I've got this in my notes, but I've made a typo. Thank you for correcting me. This is 1 minus QS. Thank you. Okay, so we have the fitnesses above, so we just need to write those in. So we're going to have 1 minus QS minus 1 on the top and 1 on the bottom, which is just minus QS. So that's just our selection differential. I'm going to put a pin in that and come back to that in a minute. So from our Fisher-Haldane-Wright equation, the format that we had above, we're now going to be thinking about things in terms of Q's rather than P's, but because it's symmetric, we can just flip everything around. Okay, so again, you can rewrite everything that's P's for everything that was in terms of P's in terms of Q's, and it doesn't change anything because, you know, it was arbitrary that we chose P over Q. So in this case, we can think about the change in one generation of allele little a, which is our recessive deleterious allele. So when I'm saying deleterious, it's not clear, it's disadvantageous. This, it turns out to be the selection coefficient times by Q, which is the frequency of that little allele, times by 1 minus Q, divided by 1 plus our selection coefficient we've just worked out times by Q. We've just worked out our selection coefficient as minus QS. So we're going to have minus Q squared S times by 1 minus Q over 1 minus Q squared S. And since our little A allele, this deleterious mutation, is rare, we can make some approximations. So Q is very small, so 1 minus Q is approximately equal to 1. And similarly, 1 minus Q squared S is also going to be approximately equal to 1. And so the change in frequency of this deleterious mutation just due to selection, we've not talked about mutation yet, the change in Q due to selection is going to be approximately equal to minus Q squared S. And that's just substituting in these approximations into this equation. So it's the change in frequency in one generation of this deleterious mutation just due to selection. And so this is negative. So this is that force pushing in one direction, saying, okay, well, it's deleterious. It's being purged from the population. So selection is trying to remove this deleterious allele from the population. Okay. Suppose it makes individuals, say, more prone to disease. Maybe they die at a younger age. Maybe they're slower and they get captured by predators, whatever it happens to be. Selection is purging that. Okay. On the other hand, we're going to have mutation. So suppose that big A allele occasionally mutates to give us this little A allele. So some error in DNA replication, for example. So if it occasionally mutates from big A to little a, we can think about the change in the frequency of this allele due to mutation alone is going to be, say, our mutation rate, which I'll call mu. Or actually, I'll call it U. Slightly easier, right? Our mutation rate U times by the frequency of our big A allele, which is P. And because little a is rare, that means that big A is very, very common. So this is just going to be approximately equal to U. So P is approximately equal to 1. So eventually we should expect the system to reach an equilibrium then, where mutation is pushing you one way and selection is pushing you the other way. And at that equilibrium, we're going to have our change in the frequency of Q due to mutation minus the change in the frequency of Q. I'm sorry, I should say Q is a frequency, the frequency of little a allele. So the change in Q due to selection is going to equal 0. And so we just substitute in the things we found above. We have U is therefore going to equal Q squared S. So it's just saying that our mutations are going to be balanced by selection. If we want to work out our frequency of this, we could leave it in this format, but this is in terms of, in terms of our mutation rate. Instead, if we want to find out an equation in terms of Q, Q can't be negative, so we just take the positive square root. We end up with Q is equal to the square root of U over S. U is our mutation rate and S is our selection coefficient. Just a reminder, Q cannot be negative. Okay, it's a pretty simple equation. What I want to do is everyone manage to copy that down. No, I can write it down. You guys to do is just like before, chat to your neighbor and work through these questions. So Tay-Sachs disease is a rare recessive genetic disorder in humans where the absence of an enzyme that breaks down fatty substances leads to death within a few years. So because it leads to death within a few years, those individuals who have Tay-Sachs disease are not able to reproduce. They die when they're children. So our selection coefficient here is equal to 1. So because it doesn't matter if those individuals are surviving for a few years, if they don't survive to reproduce, then it's whether they died at age 1 or age 10. So it doesn't matter. The selection coefficient here is just going to be 1, completely lethal. So in humans, Tay-Sachs disease has a frequency of about 2 × 10⁻⁶. That's the frequency of this disease, and it's a rare recessive genetic disorder. So I want you to answer these 2 questions. So assuming random mating, what is the frequency of the Tay-Sachs allele in the population? And then secondly, if this disease is at mutation selection balance, what do you estimate the mutation rate to be? Okay, so just use what we worked out on the previous slides. I'll put that there just so you can everyone still read the text here or not? Yeah. Okay, have a go at that. Okay, can I have a show of hands who's been able to answer these questions? Not sure. Maybe a little bit stuck, so I'll go through them. Okay. So like I said, this is a rare genetic disorder. So we think about individuals in the population that have big A, we assume as being healthy individuals. Big A, little a, and then little a. Because it's a recessive genetic disorder, like I said, it's like many of these cases are things that you just having one copy is good enough to produce the things like proteins and enzymes that you need to be able to produce to function normally. And if you don't have any copies, so you can think of this as being like 2 is normal, one of these copies is fine when you're just a carrier. So you have the same phenotype as the dominant big A allele. If you don't have any copies of that vital gene, then you're in trouble. So we're told some things so that it has that it's fully lethal. So individuals die before they reproduce. So S is equal to 1. We're thinking about selection against a deleterious allele before. So remember up here we had this when we're defining this 1 minus S here. A selection against those individuals is going to be very, very strong because they're going to have fitness 0. Okay, and we're told in humans, Tay-Sachs disease has a frequency of about 2 × 10⁻⁶. First part, we're going to be asked, assuming random mating, what is the frequency of the Tay-Sachs allele in the population? So what is the frequency of this little A allele based on the frequency of the disease? So this random mating thing here, why do we care about the random mating? Why have I said that in the question? Yeah, and why? So where does the Q squared come from? Yeah.
Q squared PQ squared comes from random.
Exactly, right. So we don't know without that assumption whether there might be some obvious sign of individuals who are heterozygous for this. And so maybe there would be choice about that. Maybe it's within sort of certain groups that might have genetic diseases because people tend to mate with people who come from similar cultural backgrounds and things like that. So we're just assuming here that there's absolutely random mating, so we don't have to worry about any of those effects, any structuring in the population, essentially. Okay, so it's not actually more complicated than it looks. As Javed just said, the frequency of individuals who are little a, so those individuals who have the disease or have the phenotype of the disease, right? Little a, they have frequency Q squared, which we're told is 2 × 10⁻⁶. And we want the frequency of the little A allele, which is just what? Yeah, it's just Q. So Q is going to be approximately equal to 1.4 times 10 to the minus 3. So what does this tell you instantly about carriers? So carriers in individuals who are heterozygous or recessive, rare recessive genetic disorders. So something that's appearing at, roughly 1 in a million people have the disease. But how many people, have the allele? A lot, right? Because the actual frequency of this allele in the population is much, much higher because of this squaring and square rooting that we're doing here. So and this is common for many, many genetic disorders that even though they're very, very rare in terms of the phenotype, if they're recessive, it's pretty difficult to produce offspring that have both. You've got to account for individuals, both parents being heterozygous, and it's relatively rare, but it's still way more common than the actual disease. But then they've still got to, only a quarter of their offspring are actually going to express the disease. So it means that a lot of recessive deleterious mutations kind of persist in the population because, it's rare, very rare for the disease to appear. And so it's very difficult to purge those recessive alleles from the population, even though you might think, oh, having both copies of them, those individuals die really quickly. Surely it would be purged from the population. Actually, they're maintained a lot of the time because heterozygotes who don't have the disease phenotype are relatively common. Okay, so the second part, we're asked if this disease is at mutation selection balance, what do you estimate the mutation rate to be? So which equation from this slide are we going to use? Yep. Okay, so we want to find out what U is. So let's work through. So I'll just start from this point here. So we have Q, as we're told, is equal to the square root of U, which is our mutation rate, over S. S is equal to what? S is equal to 1, because every individual is dying, it's told in the question. So we just have, this is the square root of U. So we have U is equal to Q squared. So we actually have this equation on the previous slide, but just kind of showing that you can work from this point here forwards, obviously. And we know what Q squared is, right? Which is just 2 times 10 to the minus 6. So actually we were given the number in the question already, simply because our S was equal to 1, right? This isn't going to be true in general, right? It's just the fact that S was equal to 1. And so our mutation rate in this case happens to be the same as the frequency of the disease in the population. That's only because of this quirk here of S being equal to 1. That works out quite nicely. Okay, a little bit more practice. This is the last thing for today. Again, I want you to think about how we might go about manipulating these things. This is slightly harder. So here we've got a wildflower. It has 2 forms, either white flowers or blue flowers. And the white flowers produce fewer seeds. We're told they produce on average 15.8 seeds relative to blue flower plants who produce 19.7 seeds on average. But they're much rarer. They only occur at a frequency of 0.00074. Okay, so rare in the population, well under 1%. And you're asked here to define the fitness of blue flower plants. Well, we're told, sorry, define the fitness of blue flower plants to be 1 and of white flower plants to be 1 minus S, because this is deleterious because they're producing fewer seeds. Okay, it's not deleterious like before and killing all the individuals. It's just there's a fitness disadvantage of being white. Maybe pollinators prefer the blue flower plants, and so they visit them more often. That kind of thing. So the first part, you want to work out what is S. So think about what it means for this to be a selection coefficient, thinking about the relative production of seeds by these different plants. So find out what is S. Secondly, assume our population is at Hardy-Weinberg equilibrium and that white flowers are caused by a recessive allele. what is the frequency of that allele? And then finally, what mutation rate would be required for the white allele to be maintained at mutation selection balance? Okay. Have a go at working through those. Yeah, let's go through the first part together and see how we get on. Okay, so we need to work out this selection coefficient. We're just given the relative, or we're given the absolute seed production of white seeds and blue seeds. So white flowers that produce white seeds, or rather, white flowers that produce seeds and blue flowers that produce seeds. We don't know about whether the seeds themselves are going to be white or blue just from that information. So let's think about the ratio of these things. So white flowers produce on average 15.8 seeds, and blue flowers produce 19.7 seeds on average. So this is just going to work out to be, I mean, that's roughly 16 over 20, so that's roughly 0.8. So this is saying that 80%, or rather the sort of the fertility of white flowered individuals is going to be roughly 80% of those that are blue flowers. So in this case, we can write down our selection coefficient as 1 minus 0.8, which is just 0.2. Why do we do 1 minus 0.8? Well, think about if the white flowers were almost identical to the blue flowers, right? They had, say, 19.6 seeds rather than 19.7. The difference between these would be very, very small, right? So our selection coefficient is 1 minus the relative fertility here. So if they were very, very similar, we would want our value of S to be very close to 0, right? Okay, so that's our selection coefficient S. The second part. So we're told to assume, we're assuming that the population is at Hardy-Weinberg equilibrium. So we can think about our P squareds, 2PQs and Q squareds, that's nice. And we're assuming that our white flowers are caused by a recessive allele. Okay, so what is the frequency of this allele? Well, we're told that the frequency of white flowered plants is going to be Q squared, is what? It's going to be 0.00074. And we want to find the frequency of this allele. This is just like the Tay-Sachs problem that we had before. So we have the frequency of Q squared. The frequency of Q is just going to be the square root of this. So it's 0.0272. So just take the square root of it. And then finally, we want to work out the mutation selection balance. So the question actually asks, what mutation rate would be required for the white allele to be maintained by this mutation selection balance? So again, we can use our Q is equal to root U over S. So Q is the frequency of this allele, S is the strength of selection, and U is our mutation rate, which we can rearrange just like before to get U is equal to S times by Q squared. And then we can just substitute in our 0.2 times by our Q squared, which is 0.00074, which is roughly 1.00015. Hopefully you got a slightly different answer to some of these things. If you did, I approximated the 0.8 at the start, just for the sake of ease of numbers. The key thing is, have you got the same process? Okay. If you are working this out, say in an exam or whatever, as long as your process is correct, even if the numbers differ slightly, that's fine. Okay, so this tells us at the mutation selection balance, what does our mutation rate need to be? Unlike the Tay-Sachs disease before, where these two things ended up, so the frequency of the disease and the mutation rate were the same. Here it's different because our S is not equal to 1. Okay? Yes.
Could you explain one again?
Yes, sure. So we're asked to find, so we're told to define the fitness of blue flower plants to be 1, and then white flower plants to be 1 minus S, and find this S. So our white flower plants, they're producing fewer seeds, right? So we can think about the relative fertility of white flowers versus blue flowers. So how many seeds do they produce relative to how many seeds do blue flowers produce? This is roughly 80% of the seeds in white flowered individuals. And so if our fitness of blue flowered individuals we're just defining to be 1, and the white flowers is going to be 1 minus S, then sort of rearranging this around, we would end up getting 0.2. Yeah. Again, if they were very, very similar, if this was like 19.6 versus 19.7, we would want the selection coefficient to be very, very small because they're very similar. And so the strength of selection is going to depend on how different their phenotypes are. If individuals are very, very similar in their phenotypes, then the strength of selection is going to be very weak, right? Okay, that's it for today. Just a quick summary, I guess. So we spent a lot of time talking about dominance today. It's kind of like one of the really fundamental aspects of genetic architecture. And we talked about how we can use the Fisher-Haldane-Wright equation to work out the how a rare allele that is either dominant or recessive and is advantageous, how that spreads into a population. How thinking about selection against an allele is just flipping that around. And so selection for a dominant allele is the same as selection against a recessive allele. And we can therefore think about sort of selection against deleterious mutations in the context of common, when a mutation is relatively rare and it's deleterious, it's the same situation as something that's rare and advantageous and dominant. I've got that around the right way. And so yeah, it's just about flipping those different ideas around. And then this idea at the end about mutation selection balance. We also talked a little bit about the additive scenario as well, where we have intermediate fitness. So yeah, that's sort of all about genetic architecture. Next week, again, reminder, there's no lecture on Monday. On Wednesday, we'll be talking about multi-locus models. So here, so far, we've been thinking about just a single genetic locus that might have 2 alleles. But obviously genomes contain many, many genetic loci. And so we're going to be thinking about what happens if we have more than one genetic locus. Now we need to think about things like recombination. And so this affects patterns of inheritance. It obviously will ultimately affect selection as well. So we're going to be talking about those multi-locus models next week. And that will be the last lecture on population genetics. And then we'll be moving on to evolutionary game theory after that. Okay. Have a good week.